Hello everyone, today we are going to learn about the cytoplasmic inheritance and we will also see the plastid inheritance in Mirabilis jalapa. See, what does the cytoplasmic inheritance mean? See, we have cell and inside the cell we have nucleus. So most of the inheritance pattern are governed by the DNA which is reside inside the nucleus. But in certain cases, it is also observed that the extranuclear DNA, that is the DNA present in the mitochondria and plastids, are also responsible for certain type of inheritance. So, the cytoplasmic inheritance is governed by the extrachromosomal or extranuclear DNA basically. Now, the evidence of the cytoplasmic inheritance was first presented by Correns in Mirabilis jalapa and Bohr in Pelargonium journey in 1908. The inheritance pattern is also known as extranuclear, extrachromosomal, or maternal inheritance. Okay, what happened in the case of cytoplasmic inheritance? Generally, the character from only one parent is transmitted to the progeny, and this one parent is usually the female parent. Mean if we have crossed a female parent having a certain character A and with male with a certain character B. So in the offspring, it should come from the both the parents and the offspring should have A and B. But in the case of cytoplasmic inheritance, what happens? If the female parent has a character A, then only the female parent character is inherited in the offspring. We are going to learn it in a bit. Okay. So the sentence means that in the cytoplasmic inheritance, the character of only one parent, usually female parent, is transmitted to the progeny. Okay. So if we do the reciprocal cross for such a offspring showing the cytoplasmic inheritance, then there would be a difference. Okay, difference in the ratio what we see, what we get in the Mendelian inheritance. Okay. Now, the gene governing the trait showing cytoplasmic inheritance are located outside the nucleus. I have already told you. Okay, all the genes which are located outside the nucleus are referred to as plasma genes, cytoplasmic genes, cytogenes, extranuclear or extrachromosomal gene. Okay, these are the different terms used to denote the extrachromosomal gene. And all the gene present in the cytoplasm of the cell is known as plasmon. Now, what are the different characteristics of the cytoplasmic inheritance? The first one is the reciprocal difference. What does this mean? I have told you that if the characters are governed by plasma genes, then the ratio which we are going to observe in the F1 or F2 generation is different from that of Mendelian inheritance pattern. So if we do the reciprocal cross here, we get a difference. Okay. Now the second one is lack of segregation. So we are going to see that the only one character from a parent is going to inherit. So there is a lack of segregation. Okay. Now third one is irregular segregation in biparental inheritance. Okay. If the character is coming from both the parent, okay, then it is known as biparental inheritance. Now the biparental inheritance give rise to irregular segregation in the F1 generation of higher plants. The next characteristic of the cytoplasmic inheritance is somatic segregation. Plasma gene generally shows the somatic segregation during mitosis. Okay. Next is mutagenesis. What happened in case of mutagenesis? Some mutagens such as ethidium bromide, they are highly specific mutagen for plasma genes only. Means ethidium bromide, if we are treating a cell with the ethidium bromide, then it will affect only the plasma genes, not the nuclear DNA. Okay. And one more characteristic is lack of association with parasite, virus or symbiont. So the plasma genes or the cytoplasmic inheritance, these are not associated with any type of parasite, virus or symbiote. Now, we are going to study about the plastid inheritance in Mirabilis jalapa. Okay, so in Mirabilis jalapa, the inheritance pattern is due to the plastid. Okay, plastid inheritance knowledge is based on the studies of chlorophyll variegation. In Mirabilis jalapa, what happened? There are three types of leaves are found. Okay, one leaf are green, completely green. Some of them are white, and some of them are variegated. Variegated means they have a shading like this. Means it also has a white portion and also has a green portion. These are all shaded. So it is written here that variegation is the presence of white or yellow spots of variable size on green background of leaves. Okay, so variegation means the shading portion. It has both the green and the white part. How does this variegation arises in the Mirabilis jalapa or 4 o'clock plant? This arises due to environmental factors, some nuclear gene or plasma gene. Okay, now let us see that when Corins made reciprocal crosses in all combination among the following, means the crosses we are going to see, and we are going to see the conclusion, what we get, what we observed. Okay, so first, leaf phenotype of the female of the branch used as female parent. Okay, this is the female parent phenotype, that is green, white and variegated. Sorry. The second is leaf phenotype of the branch used as male parent. Okay, and the phenotype of the progeny. So when green female parent is crossed with either of these three, that is green, white or variegated, observed progeny is all green. When white female parent is crossed with green, white and variegated, all the progeny become white. And here it is written variegated when crossed with green, white and variegated, then variable ratio are observed. Okay, so what we are observing here, that the progeny phenotype is same as that of female parent. Here you can see if the female parent is green, then the progeny is green. Okay, no matter what the phenotype of the male parent is. If we are taking the female parent as white, then the progeny of the phenotype progeny is all white, and in variegated the different proportion of the leaves are seen, right? So why this happened? When green plant is taken as female, all the progeny become green. White plant is taken as female, all the progeny become green, white. But when we take a variegated female parent, then different ratio of the progeny comes. Okay, this happened due to the egg cell produced from a green branch have normal proplastid, while the sperm produced on the green, white and variegated will contribute no plastid to the zygote. Okay, so what we are observing here, that the plastid which is inherited in the progeny is coming from the female parent only. Okay, this is happening due to this, and it is all the same written what I am telling you. Now you can see the picture and try to understand when white female is taken, right, and any type of male, means green, white, variegated, then the plastid is coming from the female parent now. That's why the zygote or the progeny become the white. Okay, when green female is taken, then the plastid in these leaves are green. Okay, so when the plastid is inherited in the progeny, it is becoming green. So the male parent is not contributing any of the plastid to the zygote. That's why it is happening. Okay. Now, taking the case of the variegated female, the type 1 egg may have all the white, the type 2 may have all the green, and type 3 is variegated, that is may have white and green, both the type of plastid, right? So variegated when crossed with the male parent, it may happen that the zygote is the progeny has both type of plastid, that is white and green, but after mitosis it may occur that some type obtain only the green plastids, some contain variegated and some contain only white. So this is all happening due to the plastid which is coming from the female parent only. So what is the conclusion here is that the cytoplasm is coming into the progeny from female parent only. The nucleus is coming from both the parents, but the cytoplasmic contribution is from female parent only. I hope this is all clear to you. Okay. Yeah, that's it. So thank you so much.